In this video, we will be discussing about the genetic control of the protein synthesis, which is done by the DNA and the RNA. So let's understand the difference between the DNA and the RNA. DNA is made up of genes, which is long, double-stranded and helical structure, the DNA. Whereas RNA, it is not double-stranded but a single-stranded. In DNA, it is made up of three structures that is called the nucleotide. And the nucleotide consists of the sugar, the phosphoric acid, and one of the four bases. Whereas in the RNA, it has the same things but two quite differences. The sugar here will be called the ribose sugar. And in four bases, we have adenine and guanine, that is the purines. And here we will have thymine and cytidine, these are the pyrimidines. Instead of thymidine, we will have uracil. So we will have adenine and guanine and uracil and cytidine. Now this forms the nucleotide. This is the acidic nucleotide forms. Here is one of the sample of the DNA. On the side of the deoxyribose, we will have one of the bases, for example here, and these will be attached to the other strand of the DNA with the help of hydrogen bonds. So these purple marks are hydrogen bonds. These are very loose in nature, and because these are very loose in nature, they can be split away easily. Now let's take 10 of them. These 10 of them, if we are twisting them, then it will lead to the formation of helical structure of the DNA. Now, in the synthesis of RNA, we know the formation and which components it includes. After it has formed the RNA nucleotide, it will need to be activated. For activation, it requires two enzymes, that is nucleoside mono and diphosphate kinase. These enzymes will add two more phosphoric radicals in the nucleotide, that is addition of two more phosphoric acid or radicals, which leads or has a high energy phosphoric bond. So it creates ATP, GTP, UTP and CTP. One basic thing we should know here, that adenine will bind to thymine in case of a DNA, and in case of RNA it will bind to uracil, and guanine will bind to cytidine. So in transcription, that happens in the nucleus. First, there is a DNA. This DNA has a promoter. Promoter basically means the RNA synthesis will start from here. There an enzyme will come that is called the RNA polymerase. The RNA polymerase will bind to the promoter and it leads to the initiation of RNA synthesis. This RNA polymerase will unwind everything here and it will separate the two strands of the DNA. On whichever strand it is binding, it will be termed as template strand. Now, while it is separating the two DNA strands, it will add the ATP, GTP or activated RNA nucleotide, which already has two phosphoric bonds. Now, these two phosphoric bonds will release the energy and there will be hydrogen bonds. Temporary hydrogen bonds will be made. At the end of the DNA strands, there will be chain terminating sequence. This basically means or asking the RNA polymerase to stop the RNA synthesis. Now, once the RNA synthesis has stopped and RNA has formed here, this mRNA, or this is called the mRNA or the pre-mRNA in specific, it will be gone away in the cytoplasm, and here this DNA will start to bind with the other strand of the DNA. And this was the temporary H2 bonds formed because of the RNA polymerase, and the DNA has the high affinity to bind with its own DNA strand. Now there are several types of RNA, that is 6 types: the pre-mRNA, mRNA, snRNA, tRNA, miRNA, and the rRNA. The mRNA, here's one picture of it, is formed by the process called transcription. It is present in the cytoplasm. Next is the codons. While the genes were separating, the successive three of them will be called as codons or genetic codes, which will help in the formation of the proteins. These will be termed as genetic codes or codons. The next is tRNA. tRNA has a shape of clove-like appearance, clove leaf. It has the function to transfers amino acid to the protein molecule. For example, here we have an mRNA. mRNA will have three codons here. For example, here we have AUG. So in tRNA, we will have like A will bind to U will bind to A, and G will bind to C. So the first should be UAC. As I should have mentioned here that start codon is AUG and the stop codon is UAA, UAG, and UGA. These will have the tRNA has these anticodons present. To recognize the special codons present on the mRNA, it should have some codons itself. So these codons will be termed here as anticodons. The next is rRNA. rRNA is a physical structure in the cytoplasm. It is almost 60% physical. It helps in the formation of ribosome. So there are 5 chromosomes present in the nucleus, which helps in the formation of rRNA. Now these rRNA will be collected and processed in the nucleolus. That is, inside we had a dense structure. Here they will bind with the ribosomal proteins to form granular particles that will be called as primordial structures or subunits. So let's say this is the nucleus and this is the cell. Here is the nucleolus. First rRNA is made, which will be taken inside the nucleolus. Here they will form primordial. Now these primordial subunits will go out into the cytoplasm through the nuclear pore. So it will come out here. Then these subunits will come out together. They will form a mature ribosome, functional mature ribosome. And this is how the ribosome are formed. Here is the basic structure or the function of tRNA, the rRNA, and how the tRNA has the anticodons over here. This is the codons present over the mRNA. And the next is the miRNA. In the miRNA, we will have another type of RNA that is called siRNA. The miRNA will lead to degeneration, and siRNA is short interfering or the silencing RNA. They both will lead to stop the transcription of the mRNA for the normal cellular functions. This miRNA is short, siRNA is also short, but mRNA, the size will be 21 to 23 nucleotides, and siRNA will be of 20 to 25 nucleotides, whereas the tRNA was made up of 80 nucleotides. So that's all in this video.